Hello everyone. In this lecture, we will talk about the metabolism of glycogen. Glycogen is essentially a storage form of glucose molecule, and it is very much needed to supply energy for our daily activities. Okay, who is the guy in the picture? Well, he is Usain Bolt. So in 2009, he set the world record for 100-meter sprint, which is 9.58 seconds, and it is still the fastest record today. So how can he do that? Well, number one, he is an African. Well, number two, he must have trained himself to metabolize glycogen very efficiently because glycogen provides fast energy needed in a sprint because they are composed of pure glucose molecules, and then glucose can be used in glycolysis to produce ATP under the anaerobic condition. And today we will look at glycogen, which is the energy storing molecules, and we'll look at both the synthesis and the breakdown of glycogen in detail. And here are the learning outcomes. Number one, we'll describe the structure of glycogen, and then we'll outline the synthesis and breakdown of glycogen, which is known as glycogenesis and glycogenolysis. And last but not least, we also look at the differences in between muscle and the liver, because one organ would like to keep the glycogen for itself, while the other one will release the glucose to maintain the blood sugar level. We know that for plants, starch is the major storage molecule, and starch is a polymer of glucose. Besides starch, plants also have a lot of cellulose, which is for structural support and protection. And because plants do not need to move around, they can store a large quantities of starch and cellulose. In fact, a small plant like this one is more fragile as compared to a large tree. That's because a large plant contains more carbohydrates for protection and for food, and a tree can even have a cut through and survive. So plants, because they do not need to move around, the bigger the better. But how about animals? Because animals need to move around either to hunt or to be hunted, they need storage molecules that are more compact and efficient, such as the glycogen. So why is glycogen a better choice over starch? Well, we have actually discussed this in my carbohydrate lecture. It is because glycogen is a highly branched structure. So it is composed of a highly branched homopolysaccharide. Saccharide means sugar, poly means many, while homo means you have the same type of the sugar. So when it is highly branched, you get more release of sugars per sugar per glycogen molecule as compared to the starch. And then the sugars liberated will be used in glycolysis to produce ATP for muscle contraction. And here is a very quick recap on glycogen. You should know that glucose units in glycogen are linked together by the alpha 1,4 glycosidic bonds. So what does alpha mean? It means that you have the alpha glucose, which is the alpha ionomer with their hydroxyl group at carbon number 1 pointing downward. But now all these hydroxyl group, they are already used up in the formation of the glycosidic bond. And to give a branch, for every 10 residues you have, there is a branch point which is linked by the alpha 1,6 glycosidic bond. And not only that, if a branch itself is long enough, it can also has its own branch when it's longer than 10 residues. So as a result, glycogen is a highly branched structure as compared to pure glucose. Since we use glucose all the time, so you might be asking, why not we leave glucose as monosaccharides? Doesn't it cost more energy to build them up into glycogen? After all, we are going to break them down back into glucose molecules. Well, there are two reasons, and we have studied this before. Number one, glucose is a very small molecule, so when they are carried by bloodstream into the kidney, they will be filtered through. And that means we will lose glucose through urination. A second reason is that about osmotic pressure. We know that glucose is soluble, and due to the presence of hydroxyl group, it tends to attract water. So when you have too much glucose in the cell, the osmotic pressure is building up as water is sucked into the cell, and that might cause cell lysis. So what do is that we store glucose in the storage molecules, which is glycogen. And in case of emergency, we can quickly release those sugars out again. But if you only have one chain of long polymer, you can only release limited number of sugars. But with glycogen, because it is highly branched, many sugar molecules can be released at the same time. Well, I have mentioned before that both the liver and our muscle are the main sites of glycogen storage, but they do use glycogen in a slightly different way. For muscle, it stores the largest amount of glycogen in our body because about 2% of muscle weight is glycogen. And for muscle, it is a selfish tissue because it only wants to keep glucose for its own use. So the process is glycogen will first be broken down into glucose 1-phosphate, which will be converted into G6P, and later it can join glycolysis for the production of ATP. Now, how about the liver? About 10% of its weight is glycogen, which is 5 times more than the muscle. But because liver is lighter than all your muscles, it contains less glycogen than the muscle. And for the liver, glucose is released for all other cells through the bloodstream. And therefore, the liver plays an important role in regulating our blood glucose level. Okay, we use energy all the time, but do you know how much energy we use in a day? So the value is more or less equivalent to what we consume every day. And that figure is around 2000 calories, which is around 8.4 millijoule per day. But how much energy do we have in our body? Like I mentioned before, we only have a small amount of glucose in our body, which is around 12 grams, and that can only last us for about 30 minutes. So what happens when the glucose level in the blood decreases? Our body will start to break down glycogen, and we have plenty of glycogen, which is about 450 grams that can last us for 18 hours. But remember, not all glycogen will be used up because they are more like the emergency reserve. So rather, our body will choose to utilize fat molecules, which can supply a massive amount of energy for about 2 months. And fat is about to run out. Proteins can be used to generate a big amount of energy as well. So in Africa, you will see many kids with bloated tummies. And that is actually a symptom of protein deficiency known as kwashiorkor. And if you look at the amount of energy by their weight, fats actually carry more energy than the glycogen because it stores about 2 times more of the energy than glycogen. But why then is glycogen favored as the immediate source of energy rather than fat? Well, the reason is to release energy from fat, we need oxygen because fat oxidation is a process that is aerobic, while for glycogen to convert glucose into energy or into ATP, it is an anaerobic reaction. So that anaerobic reaction is known as glycolysis. And therefore, glycogen metabolism is an important process for survival, especially in the ancient time when you are trying to run away from predators like lions or tigers. So we need to create glycogen. And whenever we see the word creation, it is genesis in Latin, and the creation of glycogen is known as glycogenesis. There are 4 key steps in glycogenesis. So it begins with glucose 6-phosphate, a very important substrate in glycolysis, followed by G1P, UDP glucose, and glycogen. And it is going to need energy investment in the form of UTP, or uridine triphosphate, a very close cousin of ATP. So this process was discovered by the Coreys. So they are actually coupled. And if you can remember, they discovered the Corey cycle, and together with this glycogenesis, they were awarded the Nobel Prize. And now let's look at a bit more details. We know that G6P is also a common substrate in glycolysis. So the first step of glycogenesis is to create a diversion by converting G6P into G1P so that glycogenesis can happen without interfering the glycolysis. And this is achieved by moving around the phosphate group from carbon number 6 into carbon number 1 by the enzyme phosphoglucomutase. And for the first step, there is no energy required. And the next thing to do is to release this phosphate group to produce UDP glucose. But to do this, we're gonna need an energized molecule, which is UTP, or uridine triphosphate. And this step is known as activation, which is the conversion of glucose. So we begin with G1P glucose 1-phosphate, which has a phosphate group and carbon number 1. And the enzyme that's going to do this activation step is UDP glucose pyrophosphorylase. So just like an ATP, UTP has got 3 phosphate groups that's attached to a pentose sugar and the base uridine. So what we're going to do is to release 2 phosphate groups from UTP. Breaking this bond will provide us sufficient energy to join these 2 molecules together, forming UDP glucose. And with that, we have made UDP glucose molecule. So this reaction is highly favorable because the release of the pyrophosphate group has a very negative delta G value. A second reason is that as pyrophosphate is released, they are hydrolyzed by the enzyme pyrophosphorylase. So to replenish for the loss of pyrophosphate, more of the UTP will be hydrolyzed and to join with glucose, forming UDP glucose. So this speeds up the reaction. And here is how the UDP glucose molecule looks like. It stores a massive amount of energy in the pyrophosphate bond. With the energy stored in the UDP glucose molecule, we can proceed to step 3, which is polymerization that is catalyzed by the enzyme glycogen synthase. So showing here is a glycogen polymer, and it always comes in 2 ends, the reducing end and the non-reducing end. For the reducing end, it means that the carbon number 1 has a free hydroxyl group, while for the non-reducing end, the carbon number 1 has no free hydroxyl group. And always remember that glucose or UDP glucose can only be added towards the non-reducing end. And with the addition of 1 glucose molecule, the glycogen has become N+1, while the UDP can be rephosphorylated to form UTP. So this final process of polymerization will just go round and round, adding a new glucose residue every time, and the glycogen molecule is getting longer. But remember, we also need a branch. So we do not want a long molecule that can only release 2 sugars from both ends, but we want a highly branched molecule that can release many sugars at the same time. So we need step 4, which is glycogen branching. In step 4, we make branches, and the enzyme we need is the amino 14-16 trans-glycosylase. Well, you can tell what it does from its name. Glycosyl means sugar, trans means transfer, while amino means a small chain. So basically, it transfers a small chain of sugar from the alpha main chain to the 1,6 branch position. So what it will do is to hydrolyze this bond. And the enzyme will keep the energy from the hydrolysis of the bond, which allows it to make a conformational change, shifting the glycosyl unit to the branch position. As you can see, the branch is now attached to carbon number 6 of 1 residue in the main chain. So this process is going to repeat over many times until we get a highly branched glycogen molecule. And then we can go back to step 3, polymerization, by extending the main chain using glycogen synthase. But here is the catch. According to a textbook, glycogen synthase can only add sugar to a polysaccharide with more than 4 residues. And due to this reason, it will need a primer known as glycogenin to kickstart the process of polymerization. A glycogenin is a protein that comes in 2 subunits, as each subunit will catalyze the addition of 8 glycosyl units to act as a primer. And therefore, you'll always see glycogenin right in the middle of a glycogen molecule. However, according to this latest study that was conducted 3 years ago, glycogen can be made without glycogenin, so you do have exceptions. We know that glycogenesis happens after a meal. That is because we have received extra glucose from the food molecule. But what happens in between meals? Well, your blood sugar level is going to drop and you need to release more glucose. But your body will not break down glycogen first, as they are reserved for emergency. Normally, our bodies will synthesize new glucose molecule by gluconeogenesis. But in emergency, for example, you are running away from a lion or a dog, then your body will break down glycogen, which is in a process known as glycogenolysis. And we'll discuss that in part 2 of this lecture.